Thank you for attending my digital talk for FNANO2020. My name is Eric. I'm a PhD student in Peter Schultz Group in Arizona State University, and I'll be presenting today about some tools we've developed for in silico design, visualization, and analysis of large DNA and RNA nanostructures. These tools are built as extensions of the oxDNA and oxRNA model. I'm sure many of you are familiar with the software package, but for those who are not, I will go through a quick one-slide introduction. Because the size and timescales relevant to DNA and RNA nanotechnology are beyond the reach of traditional fully atomistic simulations, this is a coarse-grained model which abstracts each nucleotide into a single rigid body with distinct interaction sites. The coarse-grained force field is parameterized to accurately reproduce DNA and RNA structure and biophysical properties such as helical geometry, hybridization kinetics, persistence length, and reaction to external forces. The software is GPU accelerated, allowing simulations of origami-sized particles in a matter of days on modern GPUs. Though oxDNA has been successfully employed in hundreds of papers, the surrounding software environment is not known for being user-friendly. What we present here is an attempt to remedy three main deficiencies in the oxDNA user experience: visualization, analysis, and usability. First, I would like to introduce oxView. This is a web browser-based visualization tool that natively reads oxDNA configuration and topology files. It smoothly visualizes large structures and loads trajectory files on the fly, allowing faster turnaround time for understanding your simulation outputs. In addition, this is the first editor that modifies files in the oxDNA format, allowing simulation-driven iterative modification and combining of structures. It can be found at the GitHub account on your screen. A frequently encountered problem with previous visualization options for oxDNA was the size of trajectory files. For an origami, a 2000 configuration trajectory file is often multiple gigabytes of data, which resulted in memory issues for visualizers that loaded the whole file in one go, and significantly slowed down conversions to more general purpose visualizers. To get around this, we wrote oxView to read trajectory files on the fly, leading only to a few configurations at a time. oxView also has a built-in video exporter for both trajectories and a 360-degree view of a static structure. Here, for example, I'm showing 2000 configurations from a simulation of a wireframe DNA origami. In the experimental validation of the structure, this structure appears flat and static. However, these 3D simulations remind us that these structures are not as static as they appear when stuck to an AFM grid, and in solution are quite dynamic and flexible. In the past, CAD nanostructures have been difficult to simulate using oxDNA because of the lattice-based design not exporting to a physically relevant starting structure. Untangling the helices using traditional molecular dynamics was essentially an impossible task because of the creation of topological impossibilities and a lack of directional bias toward the desired 3D shape. To get around this, oxView contains a rigid body simulator that uses a very simple physical model to reorient helices into a more reasonable geometry. Groups of particles can either be assigned manually or found using a dbScan clustering algorithm, which works quite well for structures exported from CAD Nano. To avoid topological knots, it is also possible for the user to nudge structures using the translation tools built into oxView. I should note that this video that you just saw was slightly sped up. This process actually took a couple of minutes, but the structure is now ready for final relaxation in oxDNA, which will be done in a matter of minutes to hours, as opposed to the previous method, which took days if it succeeded at all. oxView was developed using Three.js, an open-source JavaScript library for 3D visualization. Because of this, the software is easy to extend using community-built plugins, such as an STL exporter, which allows export of structures into Blender, where they can be rendered or further exported to a 3D printing mesh. Shown here on this slide is a 3D printed holiday junction, and the oxDNA model figure I showed in the first slide was a Blender render of a oxView scene. Three.js also has a WebXR plugin, which allows oxView to run on the Oculus Go headset for viewing structures in true 3D. Currently, editing is not possible in VR, but this is something we plan to work on in the future. Oh, and when I said it can load large structures, I meant very large structures. oxView is a hardware instance, which offloads a significant portion of the computation to the computer's GPU. In our tests, our computers ran out of memory before the FPS dropped below a usable level. In the video here, I show a fly-through of a crystal made of DNA origamis with about 1.2 million nucleotides. As far as we know, this is the largest simulation ever done at this resolution, and we were unable to view this structure in any other software that we tried. The next deficiency in the oxDNA software environment is a lack of general analysis tools that are structure agnostic and easy to use. To remedy this, we have written a set of Python analysis tools that perform common tasks in processing molecular simulations. These scripts will work on any oxDNA trajectory file, and they are parallelized, which helps speed up computation on large trajectory files if the user has the computational resources available. These scripts were carefully crafted to be modular and well documented, and we hope that they are useful not only for their own utility, but also to be used as inspiration for structure-specific analyses that future users would like to include for their own research. These can be found at the GitHub link on your page. Here are some examples of the outputs from these scripts. Many, including the mean structure, hydrogen bond analysis, and principal component analysis, produce overlay files that can be read by oxView as shown on these slides. These allow users to quickly and interactively visualize per nucleotide datasets. Both these analysis tools and oxView are covered in a recent bioRxiv preprint if you would like to read about them in more depth. All the code is available from the GitHub links found in the paper and in these slides. And one last thing. We are working on a web server to provide oxDNA and oxRNA simulations through a web page. We are still working on the interface and going through the beta testing phase, but we hope to have this resource open to the larger community within the next few months. We currently have an 8 GPU server hosted at ASU to run these simulations with more GPUs on the way. Further, we will be making all the code to set up this resource open source if other groups would like to streamline their internal running of oxDNA simulations. For now, this server supports simple equilibrium sampling simulations with a very basic parameter set. This is intended for experimentalists who may not have a ton of experience with command line tools to prototype their structures and get a better idea about the dynamics that their structures display in experiments. We will be implementing the previously mentioned analysis tools into this interface to facilitate their use. Unfortunately, it is not quite ready yet, but bookmark oxDNA.org so you can take advantage once it becomes publicly available. I would like to thank the rest of the team that helped make oxView possible. Misha, the original visionary who dared ask if we could do this in a web browser, Suchi and Atmik, our undergrads who implemented much of the interface, and Joachim, who visited us from Oxford for 3 months, and at that time wrote the rigid body simulator and much of the editing backend among other projects. I would like to thank our senior design capstone team, Roger, Jacob, Kai, Jeff, Suchi, and Chris, who helped create the interface for oxDNA.org. And the beta testers from Hao Yan's lab that are helping with the oxDNA.org setup while the labs are shut down. And thank you for being patient while we work out the bugs. We'd also like to thank our international network of experimental and software collaborators and our funding agencies for making these projects possible. Thank you for listening. If you have any questions, feature requests, or bug reports, feel free to send me an email or write a note on the Issues tab of the appropriate GitHub repository. I hope you enjoy the rest of your digital conference.